So if we're thinking about the things that plants need, they need light, they need water, they need CO2. We know all of this stuff already. And the way that they get all of these different things is pretty much the same for every single plant. So every plant has some sort of a root structure. Every plant has some sort of a stem structure that's going to transport the materials from the roots to the leaves, and then from the leaves to the roots. And then every plant also has their leaf structure as well. So if we dive in and we actually look at the individual structure of the leaf, it's actually really, really interesting. And most of the leaf is just air. And that kind of makes sense because if you've ever taken like raw spinach and then cooked it down in a pot, it turns from like this big fluffy ball of spinach to like it looks like nothing in the bottom. And that's because most of a leaf and most of that structure is just air in the middle. And the reason for that is because we have our stomata here, and they need to be able to intake gas and take out water vapor and all of those different things. And that gas and water vapor has to be able to travel around in different areas of the plant as well. Inside the middle of the plant, we also have something called a vascular bundle. And this bundle is made up of two different types of cells. One of them is xylem cells, and one of them is phloem cells. So we have xylem and phloem. Xylem transports things up. Phloem flows things down. Xylem is going to transport water, and really the only way to remember which one does which in my mind is because X and W are close in the alphabet and P and S are close in the alphabet. That's like the little only key that you can use to remember them. So phloem is going to be the tissue that takes the sugar from the leaves down through the stems of the plant and then stores it in the roots or wherever it needs to go. However, we have a little bit of a difference as well because in the spring, we talked about with like maple trees, that all of the sugar and sap starts to flow again, and it goes from the roots where it was stored all winter up to the tips of the branches to make leaves. So phloem can go in both directions, but xylem typically only goes in one direction because we only really want water to go up through the plant. When we're looking at our leaf as well, we have on top of it a cuticle. So that cuticle, we touched a little bit on it. It is often formed of a waxy substance on leaves, and that is to prevent evaporation and to kind of protect the rest of the leaf. And it also has epidermis cells. Just like you have epidermis, that's your skin, it's the same thing in a leaf as well with that. So when we're looking at the actual physical insides of our leaf, it is set up specifically for gas exchange and for water to flow. So those open areas are specifically for gases to go in and out. And then we have that vascular tissue, that vascular bundle, that's going to allow water and sugar to kind of go to and from where it needs to go. We talked a little bit about the stomata and how they open and close to conserve water. So when they lose water, a lot of times those stomata will close. And when they need to gain water, the stomata will open. And the reason for that is because we can think of a plant as like a giant straw. So if you have a straw and you're sucking on it, that's like the air on the outside of the leaf sucking all of the water out of the top of the plant. So basically what happens is if you take your straw and you pinch it, you try to suck through it, nothing is going to come. So when they have enough water, it's going to close the stomata and it's going to pinch the top of that straw. So when they try to suck through, nothing is going to happen. This, however, creates a lot of problems for us. And this is where we get to the last part of photosynthesis, which are different adaptations that some plants have that they've solved, which is pretty cool. So when the stomata close because they need to like maintain water, we get problems because oxygen builds up and the CO2 gets depleted. This is a huge problem. This is equivalent to us physically holding our breaths. So if we don't have enough water in the plant and they're trying to conserve water by closing their stomata, we're building up extra oxygen, which is bad for plants, and we're depleting our CO2 stores. So then the Calvin cycle can't go. becomes a problem. And then we also have the problem of our fancy gentleman Rubisco. So what happens is normally Rubisco bonds to CO2. That is the normal substrate for Rubisco. This is our enzyme that fixes carbon. Okay, this is how we build sugar. However, what happens is Rubisco, even though he's a fancy gentleman, he's a bit of a window shopper. Okay, so he loves to attach to CO2, but he loves even more to attach to oxygen, which can become a huge problem. So when oxygen concentration in our plants is really, really high, when those stomata are closed, Rubisco is going to bond to oxygen instead of CO2. So that is going to oxidize the Rubisco, and it's going to actually break down the sugars in the plants instead of building sugars. So this is where we get a process called photorespiration. So the plant is actually doing the first part of those light reactions, and then it's going into like a quasi-respiration state, because Rubisco really likes oxygen, just like a scotch more than he actually likes CO2. So when CO2 is abundant, just to kind of review that Calvin cycle, a CO2 goes in, it bonds to RuBP to make Rubisco, our sultry gentleman of the night, and then we break everything down to make a G3P molecule after we do 3 turns of this cycle. So when CO2 is abundant in normal conditions, this happens. Every once in a while an oxygen sneaks in there, but it's not a big deal. However, when oxygen levels are really, really high, what happens is oxygen will sneak in and be like, oh, Rubisco, let me show you the ways of the night. And then what actually happens is we lose and we stop the cycle, and it goes to the mitochondria. So that 2-carbon intermediate that's kind of formed from breaking down that 5-carbon into a 3 and a 2, that goes off to the mitochondria and enters respiration. Because if we remember back, acetyl-CoA is a 2-carbon molecule. So here's another 2-carbon, oh my gosh, I'm sorry, a 2-carbon molecule that then will go to the mitochondria in those plant cells and start doing respiration. Okay, so sometimes bad stuff can happen. The impact of photorespiration on plants is a pretty big deal. So it short circuits the Calvin cycle, and we lose a bunch of carbons. So we lose about 50% of the carbon that should have been fixed by the Calvin cycle. And this reduces the production of photosynthesis, which means no ATP is produced in order to generate anything in the cells, and no sugar is actually produced as well. So if we would be able to reduce the impacts of photorespiration, plants would become 50% more efficient. Photorespiration actually happens a lot more than science is led to believe. So photorespiration happens a lot, and it's not a perfect cycle. So there's a really, really, really strong selective pressure to evolve alternate carbon fixation methods. What every single plant has evolved, Rubisco, isn't very efficient. So their whole cycle kind of sucks. So some plants have operated these different systems in order to kind of thwart this photorespiration that happens in most of our C3 plants. So remember, we call them C3 because they make a 3-carbon molecule at the end of the Calvin cycle. So what C4 plants do is they actually physically separate the carbon fixation process from the Calvin cycle process. They have physical different cells in their actual plants where they separate this process, so they are able to store carbon as a 4-carbon compound. With this, you guys don't need to know how that happens. Just know that C4 plants store a 4-carbon molecule in different cells in order to like differentiate it, and they actually have a different leaf structure, and we can actually tell if a plant is a C3 plant or a C4 plant just based off of their leaf structure. And then my favorite one is a CAM plant. CAM plants only fix carbon at certain times of the day. So they are going to fix all of their carbon at night, and they do it as a 4C compound or a 4-carbon compound. And then after they've fixed all their carbon, they're able to do the Calvin cycle during the day and then make all their sugar during the day. And then at night, they do all of their carbon fixing. And then during the day, they fix all their Calvin cycle. So what happens with CAM plants is they actually store that 4-carbon compound as an acid. So if we just look a little more in depth at these different things. So a C4 plant, one of them is corn, maize, sugarcane, all of those different things, a lot of grasses. So if we think about the leaves of these different plants, they almost look to me kind of like feathers. So if you look at the leaves of one of these plants, all of the veins run parallel to each other. And when you have veins that run parallel to each other, you can actually tell that it's a C4 plant. So these things are adapted to really, really hot, dry climates. And the reason why is because they have to be able to close their stomata during the day. This is one of the reasons why corn has been so prevalent in the world as a cash crop and as a crop to feed different people, because it is adapted to really, really hot, dry environments. So kind of a cool adaptation that some of these plants have. If we delve into the inside of these leaves and their different anatomy, you can see on the side there we have a mesophyll cell, and then on the bottom we have a cell called a bundle sheath cell. So these are only going to be in C4 plants. With this, we have our CO2 coming in, and it's being fixed into something called PEP carboxylase within the mesophyll cell. And then it goes down into the bundle sheath in order to actually go through the Calvin cycle and make sugar. The reason why is because PEP carboxylase enzyme has a much higher attraction for CO2 than oxygen, so it's a lot more efficient than Rubisco. These plants are going to use both of those enzymes in order to do the same job. So it's just a little bit more of a complicated cycle, but it's a way that we can actually keep oxygen away from Rubisco. Okay, we're just trying to keep them away from each other. So we're physically separating these things where if you look on the inside, so if we look at that core, that little purple part in the middle, that's the vascular tissue where we're going to be transporting water, we're going to be transporting sugar. Those little light green cells around it are where the Rubisco is. So if we're looking at gas exchange in that whole structure, start a second period, guys. So if we're looking at gas exchange in that whole structure, we're trying to keep the oxygen away from Rubisco. Okay, that's all we're trying to do. We're just trying to keep it away from Rubisco. So if we look at a C3 plant versus a C4 plant, we can see that there's a lot less sponginess in that C4 plant. It's more filled with cells, once again, to physically separate oxygen from Rubisco. Right. My favorite ones, CAM plants. These are, I just think, super, super cool. So what happens to these is these are plants that are adapted to very, very hot, very, very dry climates, and they are going to separate their carbon fixation. So they actually have to close their stomata during the day, and then they open them at night when it's a little bit cooler and sometimes a little bit more humid. So at night, they're going to open their stomata, and then they're able to exchange carbon, they're able to do all that stuff, and they put it into storage compounds. That storage compound is an acid. That's why it's called Crochellian acid metabolism. During the day, they're going to break down those acids into carbon dioxide and put them into the Calvin cycle. So this is how succulents, so like I have a little succulent on my desk, that's how these guys actually do photosynthesis. Some cacti, and also, most importantly, in my opinion, pineapple. It is like my favorite fruit of the whole world. And there's a really, really big deal with this about pineapple, because with pineapple, you want to only pick them during certain parts of the day. And you can actually tell what time of the day your pineapple was picked based on how sweet it is. So during the night, all of these plants are going to be making and building up an acid product. So if you ever get a pineapple that tastes really sour, because we know from our taste lab, see everything building on each other, so, acid is sour taste. If you get a pineapple taste really, really sour, you know that they picked that pineapple in the morning, okay? If you get a pineapple picked at night or like later in the day after that plant has been able to turn all of that acid into sugar, the pineapple is actually going to be sweeter. Knowing this, in Hawaii, where they have like big pineapple plantations, they'll do a bunch of work in the morning on their pineapple fields, and you'll never see them out picking pineapple plants. They only pick them at night because they're going to be sweeter, or like in the later part of the day after they've turned all of that acid into sugar. It's pretty cool. So if we look at all of these different things, cacti do this, succulents do this, but also, if you guys didn't know, this is how pineapples are grown. So pineapples grow out of, like, if you imagine like cutting off the top of a pineapple plant, they literally just shove that in the ground. That forms a new plant. And then the new pineapple grows out of the middle of that plant. And it takes, get ready for this, 2 years to grow one pineapple. Never ever complain again about pineapple being an expensive fruit to buy. It takes up to 2 years to grow pineapple. Sometimes it's a little bit shorter depending on the varieties, but it takes a very, very long time. So just a little bit of a summary of these different things. We have C4 and CAM. Okay, so C4 plants separate the two steps. CAM plants separate them temporally, so different times of the day, okay, in order to retain all of the moisture in the plant. So why do we have this C3 problem to begin with? Well, Rubisco evolved when we had a really, really high carbon atmosphere. So way, way long time ago, when there was like a bunch of volcanic activity and all of this stuff, we think that Rubisco actually evolved in an atmosphere that had really, really high amounts of carbon. So back in the day, Rubisco was like awesome, and it was the key player. But now, as our atmosphere is 21% oxygen and only 0.03% carbon, it's not as efficient. However, as we're going more towards a more carbon-filled atmosphere, we are able to plant more plants and plant more trees in order to take carbon out of the atmosphere. So kind of one of those things, and it's one of those things that scientists are working on, trying to use this enzyme in order to capture carbon from the atmosphere. And they're putting the enzyme in big tanks in order to scrub carbon out of the atmosphere. So it's one of those things that's a problem for plants, but with future research, it could be good for us to actually get rid of carbon emissions. Okay, so I'm going to turn this off. I'm going to turn off recording.